So my name is Nara Jabado. I'm a clinician scientist at McGill and at its hospital, the McGill University Health Center. I have a lab at the Research Institute, and I work on brain tumors in children and in young adults.
And what is something new or surprising that you've learned lately in your work?
How COVID can affect your mental health and the mental health of a lot of people around you, and how you think that you could get along with it, but then there's nothing better than human interaction. And I think this is what we're lacking a lot these days. And my students really need interaction. I need interaction. I think I'm lucky in that I never stopped seeing people because I practice. So I went to the hospital, had to work with real people, and not everything was virtual. I think what I learned is that there are limits to how much virtual you could take. Maybe that's what I've learned.
Yeah, that's definitely a fair point to make, especially nowadays. So in terms of your patients, I assume you still have been able to see them, but I guess there has been a toll based on the school closures, and you're working with pediatric patients. So is there anything you'd like to share about seeing that overlap between their, physical health and their mental health in this current
state? I think more the mental. So for patients that are getting the pediatric population didn't suffer as much as the young adults, and the older people. So we still see patients. We had to shift a lot of our practice into kind of a mixed hybrid between virtual and in presence. There are good things to that. So there are new things that we've learned that we could manage from afar, and making people come to the hospital only when needed. But there's also still kind of fatigue. I think on the adult side is quite obvious in that a lot of the surgeries that are needed for gazillion other things, even the care of chronic diseases is truncated because there's not enough clinic space. There's not enough. We're not allowed to see those patients in person oftentimes, just because of the risk and the fact that our hospitals are so full of COVID-related restriction and infections. So I think we're just starting to see the tip of the iceberg. It's not now, it's later. And more than that, so for my patients, it's all of this angst about going back to school for their families. Should we take the risk of having them go back to school with the risk of them getting COVID? What would it do? Because they're immunocompromised, they're being treated for cancer. What would it do to them? So a lot of added anxiety that makes it hard for families, especially for families. And for those children that really would like to be with their other peers, but aren't able to make it as much as they could.
Yeah, and you definitely hope that there would be some extra accommodation in place for those particular children, but it's, I think, unclear to me whether or not those extra practices are being put in place and extra safety precautions for those children.
They are being put in place, but the moment you implement safety precaution, you implement isolation because they're treated differently. And a lot of kids just want to feel normal. And I'm speaking of kids, but think about the people, the elderly people that are in their home. They're being protected. There are a lot of safety precautions, but the protection leads also or brings also isolation with it because they're not allowed the visits, the interaction, and we thrive on social interaction, especially at different ages. This is how we become human being, and after that, how we kind of continue being human beings. So it's we're kind of keeping them safe from infection and COVID-related infection. At the same time, it's coming with other tools that we'll need to speak about. But thanks God, there is research. There are a lot of exciting stuff that is happening that takes your mind out of or help you actually make adjustment into what's happening here and there. So despite the fact that there's been a lot of restriction or whatever, there's been a lot of breakthroughs in research, some eureka moments that we've had. And the reason why I agreed to speak with you is that a lot of it is due to the genomics and to the different sequencing technologies that have made us make leaps like to no end in the understanding of deadly diseases that hopefully will become treatable in the very near future, thanks to those advances.
For sure. Thank you for sharing that. So maybe could you elaborate a little bit more about the specific work that you have been involved with?
Okay, so I was lucky in that I started using next generation sequencing from almost its inception. I'm not one of the first one, but one of the first in Canada, I would say. And this, people thought that it would not work in cancer because cancer is a complicated disease. And when we use sequencing on the high-grade glioma, which is a form of very deadly brain tumor in children, we found out that they were actually animated by a mechanism that was never thought to be here in human disease, which is mutation in the epigenome, and more specifically mutation in histone 3 genes. Histone 3 are protein that are here to help package our genetic information, but also have it expressed. It makes the fact we have the same DNA, the same sequence in every single cell that we have. However, it's how we express this genetic information that makes a cell be a heart, a fetus, or whatever. And the way to do it is the epigenome, and those histones, they package 2m10 into nothingness in the nucleus, but they also allow to open and close the areas around the DNA that needs to be transcribed when they need to be transcribed. And by identified mutation that really makes it messed up. And this was the first step. And then using a lot of those technologies that are here, the single cell transcriptome, the single cell ATAC-seq, the open chromatin, the global immunoprecipitation on chromatin marks and whatever, we showed really that the mutation we identified were making the cell be stuck in time during development. And actually more than that, it allowed us to pinpoint probably very early developmental time point during brain development, during the fetal brain development, where potentially those mutations would arise and create what I call in my jargon Peter Pan. Peter Pan age up to a point, but doesn't want to age further. So those mutations kind of stop brain development or the cells that they arise in at a given time point, and the rest of the brain development. Those cells are eternal progenitors that are cycling and cycling and cycling, and this is a new way of creating cancer. And this was all made possible based on those next generation technologies that we have at hand.
Wow, that's really incredible. I wonder if you could share, I mean, just for people like me who don't have a science background, like where did you have this hypothesis in terms of even where to look? Like what did that process look like in terms of how to narrow it down?
So we never had this hypothesis about the Peter Pan. We knew that those mutations, we knew that there was something different about those tumors in children and young adults than they were in. I work in cancer. I'm a pediatric oncologist, and what's very intriguing is what you see in terms of cancer in children and young adults are completely distinct than what you see on the more adult side. So, for example, if most cancer arise above 60 years of age. There is an exception. There are colorectal cancer, there are lung cancer, there are breast cancer, there are prostate cancer. You've heard about all of those. To see those in children is like zero. You never have this in young adults. Those are things that are acquired with time as it's the effect of our genetic, how we were born, but also how we live, and bad luck. So it's a mixture of a lot of things, and those are processes that take time. In children, you see mainly leukemias, which are super rare, cancer of the blood, that are super rare in adults. And even in the brain tumors, the entities that share the same name, and I'm insisting on name, between children and adults, when we looked more closely, and this is work that was done in my lab, we showed that really they were not animated. Even they looked like a fruit, but within fruits you have oranges, apples, or they were completely distinct from the adult side, and they were animated by much different things than the adult part. And when we digged further, and this is what the sequencing allowed us to do, is we found those mutations. And it wasn't expected. It's not that this was something that, and the fact that other people before me had worked on this histone 3 gave us a little bit of clues as to why the residue, and those were hotspot, meaning they really occurred on one area, the exact same area, everything is where they weren't random within the gene. It was at this exquisite place. And when we looked, and people had worked on this place, in other contexts, on when you're kind of learning how what I call the epigenome, how this helps express our genetic information. So I was lucky that there were some people that had done this work, and we expanded it. And those mutations that we identified, not only are they giving us a mean to better understand and hopefully soon to better treat those children with deadly cancer, but they're amazing tools to tease developmental stages, because you could use them in the other model system in order to understand, okay, what is this gene, what is this protein doing at this time point during development? When is it needed, when it's not needed? So it's beyond just those cancers that we study. It's really a tool to understand the wider picture of how our genetic information is being regulated.
Has that area been linked to any other developmental issues
Oh,
in
yes,
children?
absolutely. So again, this is the other thing that was brought about by the sequencing. There are people that identified, so the mutation that we see are only present in the tumor, okay? So the child or the adult or whatever, they don't have them in every single cell of their body. It's only the tumor that acquired them. However, there have been individuals with a lot of developmental delay and neurodegeneration, meaning diseases that make a child not very cognitively impaired and needing with a lot of needs. There are mutations in the germline, so meaning that this gene that we identified mutated in those cancer, if it is can be also mutated in the germline, and this leads to neurological impairment and cognitive issues. And I'm pretty sure there are more diseases as we're going forward, but there's something within the brain and within those histone and this specific histone 3.3 that we study that is quite important during brain development, but also as we age in potentially neurodegenerative disorders, because the epigenome is quite key to keep our memory, to keep our processes fine, and there may be other links as we're going forward.
It's very interesting. I feel like I have a lot of follow-up questions. So I guess, how is this being tracked, I guess, amongst the different people in different areas in terms of children's development that are seeing maybe similar delays or things? And is it inherited or is it just a mutation that happens?
Okay, so those mutations can be inherited, and there are specific ones. It's very rare. And the way the field is made is that we collaborate a lot. And this is something to the person that initially identified those mutations is a lady by the name of Elizabeth Broche, who works at UPenn, and her group is working actively and is communicating. All of this data is communicated in publication, and there is even there are meetings that are happening for geneticists that see these patients just to kind of compile, because it's a new entity that also is being that had no name. So people children would have this intellectual impairment, and there would be no closure. The parents wouldn't know why, because it wasn't the usual thing that would cause this type of neurodevelopmental delay. And now there is a name and there is a gene, but when there is a name and a gene, it's not to kind of label things, it's to see, can we do better? But they're not very, thanks God, they're not very frequent, they're rare, but still when they happen, you want to treat them and you want potentially to help those children have the best potential that they could and prevent the other children from acquiring them, okay? And the one that arise in the tumor, you want the tumor to stop being Peter Pan, you want Peter Pan to age and to go forward. So we're working a lot in collaboration with multiple groups. And again, those findings we have been shared. And from my lab, being the first to identify them, there's probably more than 100 or 200 labs that are working on those mutations and what their impact because of the relevance to many other diseases. And that makes a community. And the more interest there is, the better chance that more data would come and the better answers that we could have for patients, because my ultimate goal is to be able to treat and hopefully prevent in the near future.
Absolutely. Do you feel like the field of genomics is inherently about data sharing just because there's just so much information and there is that benefit of aggregating it and connecting different research together? So can you talk a little bit about that and how you're doing that in your current research with others?
I think it's not only genomics, it's everything is about sharing. I think it's what you create in It's so I'm funded by multiple different organizations, private and federal and provincial and from outside. Those are our taxpayers' money. So it's not money that is, it's given to discover, and discovery have to be shared. Otherwise, they're meaningless. It should not be kept once, because the more you have data and the more you have information, then the better you can understand, and then the better you could potentially treat and expand. The aim is not to segregate, because you have to be careful with genomic information that's super sensitive. You could have a lot of vulnerabilities that you weren't aware that you're born with. There's a movie by the name of Gattaca. It's my generation. I don't know if you've seen that movie, GATAC. It's the nucleic acid sequence in Gattaca. It's a world whereby and it's not anymore science fiction, because this is happening just by with saliva, a drop of blood. You know exactly who this person is, what's the genome, what are the sensitivities that these these people that are just born or not yet born are born with. It shouldn't be mean to segregate. At the same time, it's so much information that if it's given on small samples, it's not going to be very meaningful. You really need them to be aggregate, and then you can see pattern. It's as if when you're doing a puzzle, if it is 10,000, yeah, I'll be very nice and generous. Even if you're doing a 500 piece puzzle, if you have only one or two or three pieces of that puzzle, you're not seeing anything. You really need to have way more to see the picture and where it is. And this is what it is about data sharing. Data sharing should be really something that is almost mandated, I would say, from researchers. And it's not sharing with the organization that will take monetary advantage of that. This is not what I mean, but sharing with the organization that really will expand on the data, analyze it in a different way, confirm or infirm, and extend finding that we have. It's about that. So there is the tumor atlas, TCGA, tumor cancer genome atlas group is one of the very first that started doing that. And they have they collect their data, and they have 6 to 9 months before they really are forced to kind of put it in the public domain and for people to take advantage of it. And I think it's great, because this is what it is. They reap the benefit, but they have a time frame to kind of do it fast and do it, and then after that it's open, and it's an amazing resource. And all of our projects are put and set for data sharing. Any publication that we have, the data needs to be shared, but I want even a system whereby even before a publication that the data be out, and the data be kind of teased and used by others. There's a lot of competition, but at the same time, I think competition can be healthy and can push to faster and more meaningful discovery, and not small little pockets here and there from one investigator.
Absolutely, for sure. I guess going just back to the micro level though, with all this information and knowledge being generated, how does it then impact your patients?
So some of them have a direct impact, and others, unfortunately, it's not going to be for them. So patients that are consented for research, we explicitly say in the consent, and this is very true, that don't expect data for you. You're donating this. So what we ask them, if there is by some chance some information that's been gathered either from their germline information or from the tumor, if they want it kind of back to them, then, and they agree, we have this. And now in Canada, I think there is an obligation, there are a list of 20 or even more genes where we're almost obligated to report on them, because they have they may impact management and whatever. Obligated, no, strongly encouraged. There's it still is research. So this is the first layer. But I can tell you that we have a program that we've done within Quebec that's called Signature. It's Daniel Sinette at Saint Justine that's at the inception of this, and this is something that was very dear, but he had the drive and the power to kind of extend it, where we're sequencing every single patient with cancer at any time of his or her trajectory in the disease, diagnosis, relapse, whatever. And even though it's done under the umbrella of research, I can tell you that we're finding targets, that we're rectifying diagnoses, and we're doing actually what is going to be and should have been as of today the state of the art. It's part of what should be done upfront when you have a given diagnosis. Instead of a gene hunt going for one gene or whatever, now we have means for the same price, even for a lower price, where we could have all of the exome, all of our coding genes, analyze them and see, okay, what is the thing that's causing a genetic disease, or within a cancer, what is there something that could be more actionable? So this is something that's turning back to patients. There are patients with some genetic disorder that had no name, that just with the exome sequencing, we have a few success stories like this. Not only are we bringing the gene, the pathway, but potentially in a different therapy for those patients. So even if initially the patient signed not to have return, that this will not be of help to them, whenever we can, we try and bring it back. And if it's not for this patient per se, but it's going to be for other patients with the same disease that we're going to see in the near future.
That's really incredible and very inspiring and very, I think, hopeful for a lot of people to hear about this work being done. So thank you for sharing that. We've gone through a lot of time and it's so interesting to hear about your work. So thank you. And I think I mentioned to you, so International Day of Women and Girls in Science is coming up on February 11th. So this is a day from the United Nations to just encourage more for girls and women to enter the field of STEM. So if you had any messages that you wanted to share with anyone who's interested in this career or maybe facing, certain challenges in this field, what advice do you have to share with them?
So I'm a woman in science. I have a family of 3, and I'm very much involved. People say that potentially I'm a superwoman and whatever, and I hate this term because it's not true. I think we put our own obstacles. If we're lucky to be healthy and that our children are healthy and whatever, we should kind of go as far as our dream and our wishes. It's not because somebody is telling you should do that. It's because you want to do it, and give yourselves a mean. There are times where you're going to be investing more on personal problem, other times where it's going to be on your career. But if you want to do something, just do it. And again, failure is not a problem. It's part of, you learn from that, you adapt. And if you really want to do it again, try it again, you do it again. Otherwise, there's no success is guaranteed, but if you don't try, you never know. And I think we don't try enough. And we're our own worst enemy. We put our obstacles before we kind of go ahead and do the thing. Don't overthink it. Maybe that's my message.
It was a wonderful message. Thank you so much for sharing that. And thank you again for taking the time to speak to me and share all these incredible insights with CGEn's community. Really appreciate your time.
Thank you for your interest, and thank you for your interview.